Have you ever wondered how plants are able to grow and organize their growth in response to environmental cues? Well, one key mechanism that allows plants to do this is called polar auxin transport. Polar auxin transport is a process that involves the movement of a plant hormone called auxin in a specific direction within the plant. This process plays a crucial role in many aspects of plant growth and development, including the formation of roots, shoots, and leaves. So, how does polar auxin transport work? It all starts with the production of auxin in the young leaves and shoot tips of the plant. From there, the auxin is transported through specialized cells called parenchyma cells, located in the stem and root of the plant. Auxin is a key plant hormone that regulates growth and development. One of the defining features of auxin is its ability to move directionally within the plant. This directional movement of auxin is crucial for processes like organ formation, tropic responses, and overall plant patterning and architecture. Auxin is a key plant hormone that plays a crucial role in various developmental processes, including cell division, elongation, and differentiation. The most common form of auxin is indole 3-acetic acid IAA, a small organic molecule with a distinctive structure. Auxin acts as a signaling molecule, coordinating plant growth and development in response to various environmental cues. Auxin is essential for the proper development and functioning of plants, regulating important processes like root growth, stem elongation, and leaf formation. Understanding the role of auxin in plant biology is crucial for applications in agriculture, horticulture, and plant biotechnology. Auxin is a key plant hormone that plays a crucial role in various growth and development processes. Auxin is synthesized from the amino acid tryptophan through a series of enzymatic reactions. The main steps in auxin biosynthesis are: tryptophan is the precursor molecule. It gets converted to indole 3-pyruvic acid IPA. IPA is then converted to the major form of auxin, indole 3-acetic acid IAA. The specific auxin biosynthesis pathway can vary depending on the plant species and tissue type. Auxin biosynthesis is regulated by various factors like developmental stage, environmental cues, and hormonal signals. Understanding auxin biosynthesis is important for manipulating plant growth and development. The auxin signaling pathway is a crucial mechanism that allows plants to respond to this important phytohormone. It begins with auxin binding to its receptor, the TIR1/AFB F-box proteins, which triggers a signaling cascade. This leads to the ubiquitination and subsequent degradation of AUX/IAA transcriptional repressors. With the AUX/IAA repressors removed, transcription factors are released from inhibition and can activate downstream genes involved in diverse cellular processes. Understanding this pathway is key to unraveling how plants sense and respond to auxin signals. Auxin is a key plant hormone that regulates many aspects of plant growth and development. This slide covers the three main mechanisms by which auxin is transported within the plant. Passive diffusion allows auxin to move down its concentration gradient across cell membranes, driving short-distance transport. Active transport uses specialized proteins to move auxin against its concentration gradient, enabling long-distance transport. Polar transport is a highly regulated directional movement of auxin that is essential for establishing concentration gradients and controlling plant development. Understanding these transport mechanisms is crucial for understanding how auxin coordinates growth and responses throughout the plant. Auxin is a key plant hormone that plays a crucial role in growth and development. Auxin movement within the plant is highly regulated and directional, not random. This directional transport of auxin is known as polar auxin transport. Polar auxin transport is essential for many important plant processes like root and shoot growth, tropisms, and organ patterning. The image shows a microscopic view of a plant root tip, highlighting the regular, organized pattern of cells and the distinct regions like the root cap and meristematic zone. This intricate cellular structure and arrangement is closely tied to the directional movement of auxin that drives plant development. Auxin, a key plant hormone, moves in a specific direction from the shoot apex down towards the roots. This directional transport creates concentration gradients of auxin within the plant. Auxin moves between cells through specialized transport proteins like PIN proteins and ABSP/PGP transporters. Directional auxin transport is crucial for many important developmental processes in plants, such as root growth, shoot branching, and flower formation. Understanding the mechanisms of this polar auxin transport is key to understanding how plants coordinate their growth and development. Auxin is a key plant hormone that regulates many important developmental processes. Auxin efflux carriers are proteins that pump auxin out of plant cells, allowing it to move directionally. This directional auxin movement, or polar auxin transport, is critical for establishing auxin gradients that guide plant growth and development. Two major families of auxin efflux carriers are the PIN proteins and the ABSP/PGP transporters. PIN proteins are particularly well studied and play a central role in directing auxin flow within the plant. Understanding how these auxin efflux carriers function is key to understanding the mechanisms behind many auxin-regulated processes in plants. PIN proteins are a crucial family of proteins that facilitate the directional movement of the plant hormone auxin. These membrane-bound proteins are localized to the plasma membrane of plant cells, allowing them to transport auxin out of the cell. The polarized distribution of PIN proteins within the cell ensures that auxin is transported in specific directions, crucial for establishing the plant's architecture and development. This asymmetric distribution of PIN proteins in the root tip is essential for proper root growth and structure. The image on this slide provides a vibrant, abstract representation of plant root systems and the role of auxin influx carriers. Auxin is a key plant hormone that plays a crucial role in root development. Auxin influx carriers like AUX1 and the LAX family help transport auxin into plant cells. AUX1 is a particularly important auxin influx transporter, facilitating the uptake of auxin into cells and enabling proper root growth and development. The LAX family of proteins, including LAX1 and LAX2, also contribute to auxin influx and likely work together with AUX1 to regulate this process. Auxin influx carriers are essential for root elongation, lateral root formation, and root gravitropism, the ability of roots to respond to gravity. The activity of these auxin influx carriers is tightly regulated by various factors, including auxin levels, environmental cues, and developmental signals within the plant. The image on this slide provides a vibrant, abstract representation of plant root systems and the role of auxin influx carriers. Auxin is a key plant hormone that plays a crucial role in root development. Auxin influx carriers like AUX1 and the LAX family help transport auxin into plant cells. AUX1 is a particularly important auxin influx transporter, facilitating the uptake of auxin into cells and enabling proper root growth and development. The LAX family of proteins, including LAX1 and LAX2, also contribute to auxin influx and likely work together with AUX1 to regulate this process. Auxin influx carriers are essential for root elongation, lateral root formation, and root gravitropism, the ability of roots to respond to gravity. The activity of these auxin influx carriers is tightly regulated by various factors, including auxin levels, environmental cues, and developmental signals within the plant. The AUX/LAX family of proteins are involved in auxin influx into plant cells. They are integral membrane proteins with 12 transmembrane domains. This family plays an important role in regulating auxin transport, contributing to key developmental processes like root growth and lateral root formation. Ongoing research aims to fully understand the specific functions and mechanisms of the AUX/LAX family in auxin transport and overall plant development. The image on this slide shows a plant cell with a green arrow representing the movement of the plant hormone auxin through a PIN protein. This slide focuses on the regulation of polar auxin transport, which is the directional movement of auxin within plant tissues. There are 4 key mechanisms that regulate auxin transport. Transcriptional regulation. The expression of PIN proteins can be controlled by transcription factors. Post-translational modification. PIN proteins undergo changes like phosphorylation and ubiquitination that affect their localization and activity. Protein-protein interactions. PIN proteins interact with other proteins that influence their trafficking and stability. Environmental signals. Factors like light, gravity, and hormones can alter auxin transport patterns by affecting PIN protein localization and activity. Understanding these regulatory mechanisms is crucial for understanding how plants coordinate growth and development in response to internal and external cues. Root system architecture is shaped by polar auxin transport, which influences the branching pattern and development of lateral roots. Auxin transport regulates the outgrowth of axillary buds, promoting apical dominance and controlling the branching patterns of the shoot system. Polar auxin transport is crucial for flower development, influencing organ initiation, differentiation, and the formation of floral structures. Auxin transport contributes to fruit growth, ripening, and seed development, ensuring proper fruit maturation and seed dispersal. The slide introduces the topic of apical basal patterning in plant development. On the left, it discusses root development, where a gradient of the plant hormone auxin establishes a pattern of cell division and elongation, with the highest auxin levels at the root tip. On the right, it covers shoot development, where auxin transported from the shoot apex drives differentiation and elongation of cells, creating a distinct apical basal pattern with the most active region at the shoot apex. This apical basal patterning is a fundamental aspect of how plants develop their root and shoot structures. Tropic responses are the ways plants grow and move in response to environmental stimuli like light, gravity, and touch. Phototropism is the bending of plants towards light sources. This helps them optimize photosynthesis and grow towards the best light conditions. A gravitropism is the downward growth of roots, which anchors the plant and allows it to access water and nutrients in the soil. Thigmotropism is the response of plants to touch, which enables them to climb and support themselves, like vines wrapping around a trellis. These tropic responses are critical adaptations that allow plants to thrive and grow in their environments. Vascular tissues like xylem and phloem are critical for transporting water and nutrients throughout the plant. Polar auxin transport plays a key role in the formation and development of these vascular tissues. The cambium, a layer of cells, gives rise to new xylem and phloem tissues, and its activity is regulated by auxin gradients. In plant roots, precise auxin distribution patterns guide the formation of the central vascular cylinder, ensuring efficient nutrient and water uptake. The image on this slide shows a detailed illustration of a plant root system, with various root structures like lateral roots, root hairs, and the root cap. Lateral root formation is essential for nutrient and water uptake and is driven by polar auxin transport. Auxin gradients influence the formation of root hairs, which significantly increase the root's surface area for nutrient absorption. Auxin promotes root elongation by stimulating cell division and expansion in the root meristem. Auxin distribution is crucial for root gravitropism, allowing roots to grow downwards in response to gravity. Apical dominance is the tendency of the shoot tip to inhibit the growth of lateral buds through the production of auxin. When the shoot tip is removed, auxin levels decrease, allowing the lateral buds to activate and develop into new branches. Branching increases the plant's surface area for photosynthesis and reproduction, improving the plant's overall growth and fitness. Environmental factors like light, gravity, and stress can also influence auxin distribution and branching patterns in plants. Floral development is a complex process, with auxin playing a crucial role in establishing the floral meristem and regulating the development of individual floral organs. Polar auxin transport helps define the floral meristem identity, activating genes that control the development of sepals, petals, stamens, and carpels. As the floral meristem is established, auxin gradients and transport patterns influence the size, shape, and boundaries of the individual floral organs. The intricate auxin-driven mechanisms underlying flower development are showcased in the detailed cross-section illustration, highlighting the complexity of these structures. Fruit development is a complex process regulated by the plant hormone auxin. Auxin plays a critical role in the initiation of fruit development, promoting the transition from flower to fruit and triggering the growth and expansion of the ovary. During fruit growth and maturation, auxin influences cell division, expansion, and the accumulation of nutrients, contributing to the development of fruit size and shape. As the fruit ripens, auxin levels decline, allowing for the expression of genes responsible for the development of color, aroma, and sweetness, leading to the characteristic features of ripe fruit. Auxin also promotes seed development and the formation of the embryo, which is crucial for future plant propagation. Plants undergo various stress responses to adapt to challenging environmental conditions. One key mechanism is the redistribution of the plant hormone auxin, which regulates growth and development. Auxin transport is altered during stress, leading to changes in plant architecture and function. Plants also become more sensitive to auxin, allowing for rapid adaptation to the stressful situation. Auxin signaling pathways activate genes that trigger protective mechanisms, helping the plant survive. Auxin also promotes the formation of lateral roots, which enhances the plant's ability to take up water and nutrients from the soil. These stress responses are critical for the plant's survival and allow it to thrive even in challenging environments. Auxin, the plant growth hormone, interacts with other hormones like gibberellins, cytokinins, ethylene, and abscisic acid to regulate plant growth and development. These hormonal interactions can have synergistic effects, where auxin and other hormones work together to promote processes like stem elongation and cell division. However, auxin can also have antagonistic effects with hormones like ethylene, which promotes fruit ripening, demonstrating the complex interplay between these signaling molecules. Ultimately, the intricate network created by the interplay of these various plant hormones is what regulates the diverse aspects of plant growth and development. Polar auxin transport is a fascinating process that allows plants to grow and respond to their environment. By understanding this process, scientists can manipulate plant growth and development, which has important implications for agriculture and horticulture. Notably, auxin is a key plant hormone that regulates many important agricultural processes. Understanding how auxin is transported within the plant allows us to optimize crop growth and yield. Manipulating auxin transport pathways can influence fruit size, quality, and ripening, enhancing crop production. Auxin transport also plays a role in plant stress responses, contributing to drought and disease resistance. Developing targeted approaches to control auxin transport could lead to more efficient and sustainable agricultural practices through precision farming. Auxin transport inhibitors are chemicals that block the movement of auxin within plants. This disruption of normal auxin distribution can have significant effects on plant growth and development. I'll cover a few examples of common auxin transport inhibitors and how they are used. TIBA, NPA, and PCIB are three well-known auxin transport inhibitors. These compounds are valuable tools for researchers studying auxin transport mechanisms. They also have applications in agriculture, allowing growers to manipulate plant growth and architecture. Understanding how these inhibitors work provides insights into the complex auxin signaling pathways in plants. Identifying new auxin transporters and understanding their specific functions is an important area of research. Exploring the molecular mechanisms that regulate the activity and localization of auxin transporters will provide insights into how plants control auxin transport. Investigating the crosstalk between auxin transport and other hormonal pathways will reveal how plants coordinate different signaling networks to guide growth and development. Leveraging our knowledge of auxin transport can lead to the development of innovative agricultural technologies, such as enhancing crop yields and improving stress tolerance. Further research in these areas will help unravel the complexities of auxin transport and its impact on plant biology. Polar auxin transport is a foundational process in how plants grow and develop. It's responsible for regulating many key aspects of plant biology, from growth patterns to responses to their environment. This process is absolutely essential for plants to be able to thrive and adapt as they need to. Understanding polar auxin transport in depth gives us crucial insights into the inner workings of plants and how they function. Mastering this topic is important for advancing our knowledge of plant science and biology more broadly. So the next time you see a plant growing towards the sun or bending towards a source of water, you'll know that polar auxin transport is working behind the scenes to make it happen.